Welcome everyone to the class of marketing research and analysis. So, till now we have covered almost all the different techniques involved in marketing research. So, we have talked about majorly hypothesis testing and techniques like regression, analysis of variance and discriminant analysis, logistic regression. So, SEM, structural equation modeling, path analysis, so and factor analysis. So, today we will be doing one more technique which is called the cluster analysis. So, what is cluster analysis by the way, and how what is it similar to, and how is it different from other techniques. So, to understand that, let us first understand that cluster analysis is a technique very close to the factor analysis, right. So, as factor analysis you have done, and you know that in factor analysis the basic objective of factor analysis is to do data summarization and data reduction, right. Similarly, cluster analysis helps you to create groups among similar objects, right. So, the only difference between the cluster and the factor is that in factor analysis you are trying the basis of grouping was the variables. That means, on basis of the various variables you are creating a data reduction technique, and thus a few of the a large number of the variables were being grouped into a few ones, right. But here the objective in cluster analysis is not to group the variables, but rather the objective is to group the cases, means the respondents. So, can we that means the objective is to see, can we group or create groups out of a large number of respondents into a few numbers, so that we can say, well, a particular group, let us say one particular group has a particular kind of behavior and another group has another kind of behavior. So, accordingly maybe a marketer can use that knowledge for his or own benefit. Okay. So, let us see, let us go get into it now. So, cluster analysis is a group of multivariate technique, right, whose primary purpose is to group objects based on the characteristics they possess, right. So, it is basically an interdependence technique, again like factor analysis it is an interdependence technique. So, there is no dependent and independent variable as such, right. It is a means of grouping records based upon attributes that make them similar. So, on what basis, on what attributes, what are the attributes that make similarity, creates a similarity among the respondents is what is of interest to the researcher. If plotted geometrically, the objects within the clusters will be close together, while the distance between the clusters will be farther apart. That means, if there are two clusters, let us say this is cluster 1 and cluster 2. So, what we are saying here is the the data points within the clusters are as homogeneous as possible, and two different clusters are as far as possible. So, there is a clear cut distinction, right. So, that means it says homogeneous within the cluster and heterogeneous between, right. Cluster analysis is also called classification analysis or numerical taxonomy. Now, if you have read in childhood when you were doing classes of biology and zoology, you are studying about different species, right, how different animals can be grouped into different species or different species basically, right. So, we some were into, like for example reptiles, some were into homo sapiens, for example human being is a homo sapien, some was getting into fungi, somebody into algae. So, there were different species, right, of plant and animals. So, according to the characteristics we were classifying them. So, how was it done? This was done on the basis of, with the help of such techniques like cluster analysis, right. So, you see, for example, this is a just an example where it says, if you can see, there are estimated number of clusters is 4, all different, shown different colors. So, this is a slightly greenish color, this is sorry light blue, this is dark blue, green and red. Now, each one is showing, depicting a particular cluster. Now, if you can see, this cluster is the farthest from the other clusters, but these 3 clusters are very close to each other, right. So, this is just an example to depict how clusters are formed. So, you can say, if you take it in, like for example the species what I said. So, this could be humans and this could be like the fungi, algae and some other kind of plant or something, right. A ideal clustering is basically like this, it seems to be, but nothing comes so clear in real life, right. So, basically the data points are spread up in this manner in a real life. So, practical it says ideal, this is ideal and this is practical. So, when you are doing a clustering, the data points if it is so clearly distinctly separated, then nothing like it, but it does not happen in real life. So, sometimes that is why you see some of the clusters will show some superimposition, some parts of it will try to jumble upon each other, right. So, this is why, because this you see this data might are so close. So, it might create a situation like this. So, clusters are extremely close to each other. So, the difference between cluster analysis and factor analysis, it is a classification technique, this is a dimension reduction technique, factor analysis. The basis is the grouping is based on the distance. Now, this is very important. Grouping is based on the patterns of variation using the correlation. So, in the factor analysis, the major objective was to create the groups of the variables on basis of the correlation among them. But cluster analysis does not take the correlation, it takes the distance between the variables. Okay. Statistics used is the dendrogram. It is dendrogram is a diagrammatical representation, cluster, centroid and agglomeration schedule. I will show you what they are. This helps you to separate or create the clusters. Here we used, if you remember, eigenvalue, factor loading, which was nothing but the correlation between the variable and the factor score and commonality. So, these were some of the things that differentiates the cluster and the factor, and this is basically is done on cases or respondents. This is done on the variables. This is the most important, simplest and the important difference. Similarly, cluster analysis versus discriminant. If you remember, discriminant analysis was a technique which was used. The dependent variable was a categorical variable, right. So, you wanted to take a decision whether somebody should be allowed into the class or not permitted into the class, somebody should be given a bank loan or not a bank loan. So, there we were using discriminant analysis, right. So, what is the difference between these two? Why we are saying this? Now, in case of a cluster analysis, it does not require any prior information. It is a actually what happens is cluster information analysis is a subjective, right, study, right, is a subjective measure basically. So, any prior information about the group or membership is not required, but in discriminant analysis, prior knowledge of the cluster or group membership for each object or case included to develop the classification rule is required. For example, when you are discriminating, right, the groups, for example, there are two groups. So, in order to discriminate these two groups, you need very clear information about the groups. Okay. This is an, as I had said, it is an interdependence technique and this is a dependence technique, right. Why? Because there is a dependent variable which is categorical and there is one or more independent variables which are continuous. Okay. For example, I said pass fail, right, and this is on hours of study, IQ of the student, etc. Where is it applied? In the field of psychiatry, where the characterization of patients on the basis of clusters of symptoms can be useful in the identification of an appropriate form of therapy. See, when you talk about the mental health, for example, there are different kinds of patients. Everyone cannot be treated similar, right, or same. So, in such a condition, it helps us to decide at what level of mental condition is the patient lying. So, that helps us to give different kinds of medicines, right. Biology, for example, to group the genes that have similar functions. Some genes are responsible for your happiness, some genes are responsible for somebody is being aggressive, right. So, what are the genes that are responsible for these? So, that helps you to identify through a cluster analysis. The application of cluster analysis has improved like anything today, right. In World Wide Web consists of billions of web pages, for example, you see clustering can be used to group. This results into small number of clusters, each of which captures a particular aspect of the query. For example, somebody is interested to know about the what is there under the sea. So anything linked to sea and all that can be clustered into one group. Something about the sky, the universe, the Milky Way, so that is another group. Or sometimes you can find out if sea and reptiles could be taken together, maybe that is a cluster. So such kind of clusters can be formed, right. Understanding the Earth's climate requires finding patterns in the atmosphere and ocean. Cluster analysis has been applied to find patterns in the atmospheric pressure of polar regions and areas of the ocean that have a significant impact on land climate. So such kind of applications are very much there, right. Similarly, cluster analysis has been largely used in marketing, right. First of all, it is used for segmenting the market. Now, on what basis do you segment? What is segmentation? To divide the market into several parts, so that each part has got some some measurable value. Okay. Example, consumers may be clustered on the basis of benefits sought from the purchase of a product or service. For example, you see sometimes some customers are extremely customers are divided in such pattern. For example, some are called to be innovators, one who try the product for the very first time and they want to test it initially. Some people are those who want to check the product after only it has been used. So what is the kind of pattern or behavior of these people, right. So what factors affect them? So these kind of benefits sought and things help in segmentation, which can be done through cluster analysis. Understanding buyer behavior. Cluster analysis can be used to identify homogeneous group of buyers, then the buying behavior of each group may be examined separately. That means, suppose I have created two clusters, let us say 1 and 2. So cluster 1 has got certain behavioral characteristics. For example, we have taken characteristics like happiness, sports, sportsmanship, right. For example, team person. Now, what is the score that the cluster 1 people are getting? What is the score the cluster 2 people are getting? So accordingly, we can see which cluster is a better cluster for us. Identifying new product opportunities. By clustering brands and products, competitive sets within the market can be determined. Obviously, so when you can create different clusters, obviously then you can target which cluster you are you like more and would be more beneficial for the company. Sometimes test markets are selected on basis of clusters by grouping the cities into homogeneous clusters. So it is possible to select comparable cities to test various marketing strategies. So what you can do is you can create a cluster of cities. So any city that falls in a particular cluster are similar, right. So if I want to select, let us say my target segment falls into cluster 1, let us say, and in cluster 1 there are 5 cities. So I can select any one of them and use it as my test market. Okay. Finally, reducing data, for example, as you are doing in factor analysis, you can do it in cluster analysis too. How does it work? That is most important. The primary objective is to define the structure of the data by placing the most similar observations into groups, right. So it is a matter of similarity or dissimilarity, right. So how similar these observations are. To accomplish this task, we should know how do we measure similarity, how do you measure, right. How do you form clusters and how many groups should you form, right. So how many clusters, how do we measure the similarity and how do we then understand this each cluster, okay. So similarity represents the degree of correspondence among objects across all the characteristics used. Please remember, this is what I was trying to say. There are two ways you can understand the similarity, right. One through the correlation measures which you are doing in factor analysis. So in the factor analysis we said the variables which are very close to each other or have a high correlation would fall into one factor. But in the case of cluster analysis, we are not taking the correlation measures, right. We are often linked to the related with the distance measure. Most often used as a measure of similarity with higher values representing greater dissimilarity, distance between cases, not similarity. That means what? If the distance between two clusters, let us say cluster 1, cluster 2 and let us say this is cluster 3. Now this is 1, 2, 3. So higher the distance, that means farther the two clusters. That means in a layman's term you can understand that cluster 1 and cluster 2 are more different or much more separate to each other than in comparison to cluster 1 and cluster 3. So, this is a closer pair and this is much farther from each other. Look at this similarity measure. Now, both graph, if you can see, if you look at this, right, the pattern of the data. So, it rises then falls more, rises falls more, right. So, same thing is happening in both the cases. So, this is if you check it in terms of correlation, if you see both are having a correlation of 1, right. So, both graph have the same r is equal to 1, the correlation is equal to 1, but if you look at the distance between the points, now you can very easily see that here in this case there is a much farther distance, that means the data points are at a much farther from each other, right. So, that is what it says, graph 1 represents higher level of similarity, right. So, look at this for example, now this is 2 lines. So, this A and C, if you look at the pattern of the data, if I put in a data they are very similar to each other, but if I now put in a third point, right, let us say this one, the B line, and now you can see that the pattern of the data is different. Now, if I measure, let us say this point and this point and this point and this point, you see now the distance is becoming different. At this point it is very less, but at this point it is very high, or this point it is very high. So, now A and B and C are different from each other, right. So, the distance is different from each other. So, what basically we are doing in a cluster analysis is to measure the distance. Basically we want to say how far is one cluster from the other, or how far is one variable from the other, or how far is one data point from the other. This is what we want to see, not the correlation, not the trend. Correlation is basically the trend, the pattern. So, we are not talking about the pattern, we are talking about the actual distance, right, and we measure here the Euclidean distance. The Euclidean distance is referred to as the straight line, right, and there are some distance measures. For example, Euclidean distance, which is the straight line distance. Squared Euclidean distance is the sum of the squared differences, without taking the square root, right. So, for example, x we say equation of straight line is x2 minus x1 square plus y2 minus y1 square, right. So, root over. Now, if you just remove the square root, then this is the squared Euclidean distance, right. CT block distance, it uses the sum of the absolute differences of the variables, right. I will show you in the next slide. Chebyshev is the maximum of the absolute difference in the clustering variables values, and Malebis. This is used when there is a high degree of correlation among the variables. In order to avoid that situation, whenever you have high correlation among the variables, at that condition the Malebis distance is a very right measure to be used, right. To it basically what it does is it standardizes, it is a generalized distance measure that accounts for the correlations among variables in a way that weights each variable equally. That what it basically does, it standardizes the variables, okay, the values. Now, this is what we are talking about, the Euclidean distance, the CT block distance. Now, this CT block distance is, as you can see, you can read it, it uses the sum of the absolute differences of the variables. Now, this, right, plus this, right, is the CT block distance. The Chebyshev distance is this part. So, there are different distance measures, okay. Now, let us take this example. A marketing researcher wishes to determine tourism segments in a wellness destination based on patterns of tourist emotions. Now, on basis of the emotion he wants to create segments towards the destination. A small sample of 7 respondents is selected on as a pilot test, right, and cluster analysis is applied. So, 2 measures of emotions V1 and V2, right, and 7 respondents are there and the data are given to you, right. The emotions were measured on each respondent on a 0 to 10 scale, okay. Now, when we represent the same data V1 and V2, you see, so the first case was 3 2 for example. So, A is 3 2, right. So, B is 4 5, right. So, we have plotted it, right. So, but from here we can't say anything much, okay. Now, what is done is how do we measure the similarity, that was one question we had in our mind, right, how do we measure the similarity. So, to do that, this is the formula we use. So, for example, for A and B, so I have put that same table here, I have copied it here. See A and B. Now the distances, how do I measure? Now V1A, that means this, right, minus this, okay, square, plus this minus this square, right. So this is 3.6162. Similarly for A and C, you can do A and D, A and E, A and F, A and G, you can do everything, right. So 3.162 we have found for A, B and A, or A and B, same thing, right. Now for let us say A and C, so 3 minus 4, 1 square, so plus 2 minus 7, 5 square. So 1 square plus 5 square, 26. So root over of 26 is equal to 5.099. So similarly you can do it for the rest, right. So every value has to be written. How do we form the clusters? Identify the two most similar observations not already in the same cluster and combine them. So what you do is, first once you have these values, after that you start doing your clustering. So first find the similar clusters and try to combine them, right, and you continuously do it till you reach the single cluster. This process, right, single cluster, this process is termed as hierarchical procedure. Hierarchical because it is in an order, right, because it moves in a stepwise fashion to form an entire range of cluster solutions. It is also an agglomerative method because clusters are formed by combining existing clusters. So that is why it is an agglomeration, right. So it could be agglomerative or divisive, we will see, right. So how do you do it? So we have found out these values, the initial solution. Now this is the pair we have noted, key 1.414, so E and F, so E and G is this, C and D, B and C, B and E, A and B. So these are the different values, right. Now this is the first initial solution, A, B, C, D, E, F, G. There is no combination, there is nothing, no clusters have been clubbed till now. Now first we will start, how do we do it? Now we will first club the closest one. So this is the, if you see it is put in order, right. So E and F, first we are clubbing E and F, right, and then how many clusters are left? 1, 2, 3, 4, 5, 6, right. So this value remains the same. Then next is A, B, C, D, E, F and G. So E, F and G we have clubbed. So how do you do here? Now you see, now this value you must be thinking how this value has come. Now to do that, what you do is E and F, how much? 1.414, E and G 2. So let us add these two, right, 1.414 plus 2 plus F and G, right. So F and G you can measure it here. So F and G for example, so 7 minus 6 square, 1 square, plus 7 minus 4 square, 3 square. So 1 square plus 3 square, which is 9 plus 1, 10. So root over of 10 is 3 point something. So you see, right, 3.162. So now this plus let us say this 3.162 kind of a value. So if you add it here and divide it by 3, then you will get this score. So rest you can do it for the others also. So now we have clubbed, and you see each, by clubbing each step, now you have come across to a point where finally you have got one cluster. So in steps 1, 2, 3 and 4 the overall similarity measure does not change substantially, right, 1, 2, 3 and 4, which indicates that we are forming other clusters which essentially the same heterogeneity of the existing clusters. Okay. When we get to step 5, we see a large increase, step 5, right. So there is a large increase, okay. This indicates that joining clusters B, C, D, B, C and D, this one, right, B, C, D and E, F, G, right, resulted in a single cluster that was markedly less homogeneous. Okay, you just understand that this is how the clusters are formed. Okay. Therefore, how many groups do we form? The question comes. Therefore, the 3 cluster solution of step 4 seems the most appropriate, right. Let us see the for example here. So if you look at these values, now where is the change maximum? Now here there is hardly any this change, there is a change, yes, there is a big change here also, but there is hardly any change, slight change. Now if you go from the bottom, right, let us say you go from the bottom, so there is some change here, there is change here also, but there is slight change and this is almost no change, rather it is increasing. So, how many clusters have to be formed, we can think accordingly. So, if you see step 4 seems the most appropriate for a final cluster solution with 2 equally sized clusters B, C, D and E, F, G and a single outlying observation A. So, this is what we are talking about B, C, D, E, F, G and a cluster A. So, on the 4th step, in the 4th step this part, right, this is the we are talking about this part. So, this is how it looks like, right, B, C, D, E, F, G and A is alone, right. What is dendrogram? It is a graphical representation, a tree graph it said, of the results in a hierarchical procedure. So, how it has been clubbed you can see here. So, starting with each object as a separate cluster, the dendrogram shows graphically how the clusters are combined at each step of the procedure until all are contained in a single cluster. So, first you add one, then you go on adding to the next. So, slowly you go on adding till you get the 6th cluster, right. So, let us say this is the 1, then with 1 and 2 is combined, then after that 3 and 4 are combined, then 3 and 4 are combined with 1 and 2, right, and then this total 5 is combined with 6. So, it goes like this. How do you derive the clusters? There are a number of different methods that can be used to carry out a cluster analysis to understand how many clusters should be made. One is the hierarchical cluster analysis, the other is a non-hierarchical cluster analysis and the third is a combination of both. So, hierarchical cluster analysis I will just brief you and then we will wind up here. In the next lecture we will carry on from here. In the hierarchical cluster analysis, as you have seen, it helps you to indicate the number of clusters that need to be formed, okay. And in the non-hierarchical cluster, which is basically if you see any test it is shown as the K-means cluster, right, is used to identify and analyze the behavior of each cluster, okay. So, when these two methods are used together, A and B, this and this are used together, it is a combination of both. This is a very it gives a very apt solution for any researcher to derive the number of clusters and understand them very critically, right. So, what we will do is we will continue with this lecture in the next this topic in the next lecture, and we will also see how it is being done in the SPSS, right. So, thanks for today.